out in a different camera this morning, so I want to try it. Alright, looks nice. Everything's in focus. Good. Well, I can post experiment 4 if you really want to do it. It won't be worth any points, but go for it. Alright. Okay, so let's start from the beginning. So we have our cell here, and okay, we've got a cell here, and we've got our nucleus, we've got our chromosome, a couple of chromosomes in this nucleus. Okay, and we have a gene, and this gene that we're interested in is going to be turned on. So what do we call it when we say a gene has been turned on? What's a more correct term?
Express. Gene express.
Right, gene expression. So we're talking about gene expression here. Okay, so we got gene expression. So what's the first step in gene expression? Oh, louder? Yes, transcription, right? So we're going to get transcription. Alright, so I'm going to erase this chromosome. So we're going to get transcription. Okay, so now we have an RNA. Let me see how this looks. Okay, now we have an RNA. And, what needs to happen next? So what's, oh yeah. So what's this RNA? Describe it. So this is pre-mRNA, right? So pre-mRNA. Okay, so what's pre-mRNA? What are my boxes and lines?
Exons. Introns and exons.
Exactly. A couple of people who answered. So boxes are exons. And these are the introns. Okay, what's the difference between the two?
The introns are the non-coding parts.
Okay, so the exons are? The introns are non-coding, the exons are?
Coding.
Right, the exons code for the protein, right? So what has to happen to this transcript? Oh. What's the next step?
Splicing.
Splicing. And that does what? Right, it removes the introns. So now all we have left are the exons. Right, so we've gotten splicing. And now we just have that. So backing up, transcription, what enzyme is required for transcription?
RNA polymerase.
Exactly. And for splicing? I believe somebody... I think you said it, your audio is really, really weak. Spliceosome? Right, the spliceosome, okay? And it does the splicing, and now this can exit the nucleus. So we've got the transcript in the nucleus now, so it's now our mRNA, or mature mRNA. Now what happens?
Translation.
Right, so now we get translation. So for translation, what needs to happen?
The mRNA needs to enter.
Yeah, enter. Nope. Right, it goes into the ribosome, and with the help of what molecule?
ribosome.
Yeah.
tRNA.
Right, the tRNA, we're going to make a protein, right? So we're going to get protein synthesis. Right? So here's our ribosome with the help of the tRNAs coming in. And what is the job of the tRNA?
It brings the amino acids to the ribosome.
Right, and well, do you know what order to put them in?
It matches up with the codons.
Right.
And brings the according, the matching amino acid.
Exactly. So remember, the tRNA has the amino acid at one point and the anticodon below, right? So for AUG, you would have UAC, right? So that anticodon would base pair with the codon and bring in the proper amino acid. Okay, so that's how it's happening in our cells. Now, if this gene is turned on in every cell, what kind of promoter would it have? Maybe it's hexokinase. Right? Hexokinase, I think, is expressed in every cell in our body. What kind of promoter would you predict that hexokinase has?
Constitutive?
Right, constitutive, or always on. Exactly. Or what if it's only expressed in one cell type?
Conditional.
Right, it would be a conditional promoter, right? And whatever condition is required would turn on that gene. Or in some cases, turn it off, right? Maybe it's on and it needs to be turned off for some reason. Okay, a condition is met and is turned off. Right, exactly. So that's the basics of gene expression in a eukaryote. Now, okay, so now let's go to coronavirus. And specifically, of course, COVID-19. Okay, so what type of genome do we have? What type of genome do the eukaryotes have?
Genome?
Genomic, but what type of nucleic acid?
AGC?
Right, so DNA, exactly. So our genome is DNA. Is it single-stranded or double-stranded?
Double.
Right, it's double-stranded, right? It's a double-stranded. Each chromosome has two strands. They're double-stranded, and it's DNA. So we have a double-stranded DNA genome. Okay, the coronavirus does not. The coronavirus is a single-stranded RNA virus. Okay, so in the capsid, so we got the viral capsid, which looks nothing like this, but we'll pretend like it. It has a single-stranded RNA genome. Okay. So when it affects this cell, it kind of skips this part. Okay. So let's draw a new cell. I'm going to see how I can do this. So this is eukaryote. Now we're going to talk about the coronavirus. But we've got to start with a eukaryotic cell. Okay, with our own genome. So we'll have our own DNA genome. Okay, and there will be 23 pairs of chromosomes in that nucleus. And now we've got the coronavirus. So the coronavirus comes along and it has, and I don't know how many copies of the RNA it has, but it probably has several. in the capsid. So this circular thing would be the capsid, which is the protein coat that surrounds and protects the RNA genome. And then it has these proteins that stick out, and this is spike, or infamous spike protein. Okay, and it just so happens, coincidentally, that that spike protein can bind to a receptor on the eukaryotic cell. Okay, so it can bind there, and when that does, the virus is invited to become part of the cell. Okay, so it's hijacking this ability to bind to this receptor, and that receptor says, okay, you can bond to this membrane and release your RNA. Okay, so now the RNA is in the cytoplasm. So there's an RNA in the cytoplasm, and one of the genes here is an RNA-dependent RNA polymerase. Okay, so it's RNA-dependent RNA polymerase. So what do you think that does? What does an RNA-dependent RNA polymerase do? Anybody want to guess? Or does anybody know?
Is it extracting RNA to DNA?
Not quite. Okay, so it's RNA dependent. So what does that mean? Well, first, let's back up. We all know, what do RNA polymerases make?
They replicate the RNA.
Okay, so this whole thing replicates the RNA, but specifically RNA polymerase. What does it do? We just talked about it back here, right? What molecules does it make? It says it. Its name. Right. Okay. In the chat. mRNA. Yes. RNA polymerase makes RNA. And in the case here, it uses DNA as its template. So this is a DNA-dependent RNA polymerase. So here we have an RNA-dependent one. So what do you think this template is for this RNA polymerase? What is it? It broke up.
RNA.
Yes, RNA, right? So it has the enzyme to make copies of this RNA. Okay, so it brings in the enzyme that will copy its genome. Okay, and that is replication. Okay, because we're making the same molecule. Just like when we replicate DNA, we make DNA. When we replicate RNA, we make more RNA. Okay, because we started with an RNA template. If we start with DNA to RNA, it's transcription. So we have this molecule, this genome in the cell. It's going to use this cell's ribosomes, tRNAs, and all the nutrients to make this enzyme to make more copies of the genome. Okay, so it's going to actually use, sorry, what did I just say? Right, it's going to use RNA, TNA, and ribosomes to translate that gene so that it can then make more copies of the genome. Okay, so then we get lots of copies of the genome. It also uses the RNA polymerase, I'm sorry, the cell's ribosomes and tRNAs to make other proteins, and you'll see those later on when I show you the genome browser. But there's about a dozen proteins, including spike and all the other parts of the capsid and some other enzymes that are translated in the cytoplasm to make more virus. So ultimately, you make more virus components, and these genomes get encapsulated with more virus, with spike proteins and other proteins, and eventually the cell bursts. and you get new virus out in to infect more cells in the body, or if you cough, this person coughs on something else, those viruses go to another person and infect another person. Okay, any questions about this? So we go from here to replication to new virus, and then the cycle repeats. Okay. So basically what the virus is doing is shortcutting this part. We don't need to do transcription. We're just going to make more copies of our RNA genome and translate that, and then have make more virus particles. Questions about that?
Why does it skip transcription?
Oh, because there's never DNA. This doesn't, as far as I understand, the RNA genome is copied directly back into RNA. Okay, there's no cDNA here. That's a different virus, which is also a single-stranded RNA virus, but it's a retrovirus. We talked about it early in class, right? And that one has a very different lifestyle. So, now let's talk about the Pfizer and Moderna vaccines. So this is the natural way that things happen. And now we'll go another way. I'm going to make my nucleus smaller so I can use the same cell. There. So the vaccines. And this is just the Pfizer and Moderna. Okay, both of them are RNAs. Okay, they're RNAs in a lipid droplet. And that droplet protects the RNA, but also allows it to go into the membrane. Right? So you have a lipid droplet with an mRNA to spike. Okay, so we've got spike protein here, or spike, sorry, the mRNA to spike. Okay, because spike is what is going to be eliciting the antibody response, the immune response. So they put in the mRNA for spike. Okay, and we're not generating, and we are not looking for antibodies to the RNA. We need antibodies to the protein. Okay, so this lipid droplet fuses with the membrane. Okay, and spike protein, or sorry, spike RNA goes into the cell. So how do we get spike protein? This is RNA in the shot that they stick in your muscle, but we need the protein. So what's happening? How can we get a protein from our mRNA?
You have to translate it?
Exactly. So we're basically doing like the virus and asking the cell to translate this mRNA for us. Right? And so we do that. We get translation. And we get spike protein. And that gets put on the surface of the cell. Okay, and now we can go fishing for antibodies. So that's how it works. So we just are injecting the mRNA into the spike, into the muscle. That lipid lets it bind to the lipid membrane, the RNA goes in, and then we use the translation machinery of the cell to make spike. And we can make a lot of spike, a lot more than could potentially go into a more traditional vaccine. The difference between the two, or any questions about this before I move on?
So the vaccine, we're actually making antibodies for the spike and not the actual RNA inside the virus?
No, we're not making the antibody. We're making the actual viral spike protein.
Oh.
Right. The body's going to make the antibody. We'll come back to that.
Okay.
But this is the viral mRNA. We've just taken the one gene out, and we'll see this later in class. We've taken the one gene out. You can make these RNAs really simply, or fairly simply, in great big vats of bacteria. Clean it up, encapsulate it, and then inject it so that it goes into the cell, and we translate it, and we get the actual spike protein. That's the only thing, so we don't get sick, right? It's not the whole virus. It's simply one mRNA. Good question. Any other questions?
Yeah, so that spike, is that the same spike that you're pointing to in the, yeah, right there?
Yep, same one. So this is just from the natural virus. We've made this one through isolating the RNA, but the protein is the same.
RNA.
So when
So with the
sorry, go ahead.
sorry, you can go. So with the spike protein going on the surface of the cell, that makes it so the coronavirus can't bind and infuse with the.
No, we'll talk about that in a minute. We're not going to get into a lot of detail of immunology, but as cells do their job, do their functions, they routinely put proteins on their surface for the immune system to check. Okay, and if it's a normal cell protein, the immune system ignores it. But if it's a foreign protein like this spike, if there's an antibody that recognizes spike, then that anti-spike antibody will trigger the immune system. Okay, but this is a random event, and we'll come back to that. Okay? Somebody else had a question.
No, she asked the same question I asked.
Oh, good. Okay. Right. So cells just routinely put different proteins on their surfaces, even though that's not where they really belong, for the immune system to be able to check, is this cell making only what it should be doing, or has it been invaded? Okay? It's a pretty complicated system, so we won't get into it. But definitely, if you get a chance, take some immunology. It's quite fascinating. Right, so this antibody. We're not going to talk too much about it, but just so you kind of understand how the immunity works. These antibodies are generated randomly in our cells, okay? In the cells of the bone marrow that make antibodies, and the B cells, they randomly rearrange the chromosome so that they make unique antibodies. Okay, and there are millions of these things in our bodies. Okay, just floating around in the blood and in the lymph, and they're randomly generated. Now you may think, well, if you're randomly generating antibodies, you might make an antibody against self, and that happens. And those get screened out during the whole process of antibody development. So if it recognizes self, it gets killed off. That cell will get killed off, and others get to live and have the antibodies produced. And so randomly, people will have this antibody. And if this presented with its antigen, okay, so the spike protein is the antigen, then it triggers the immune system to go into full function. Okay, and then by going into full function, it goes into mixed memory cells so that this particular antibody will stay around longer. You get a booster, so the immune system sees it twice and becomes really, trained that this is a foreign protein, and we need to basically tell this cell to die. it'll trigger the cell to it'll either be triggered to do what's called programmed cell death, or it'll get engulfed by another cell and destroyed. Okay. But that's how we elicit immune responses with these types of vaccines. Now, like the AstraZeneca and the Johnson & Johnson, they're the more traditional vaccine where it's either dead virus or just a protein. So they're injecting the protein directly. And that's the way it's always been done. This is the first time the mRNA virus vaccines have ever been used. And as you see, they came out more quickly. These are faster things to develop. It takes longer to develop the more traditional type, and they also are going to be more responsive, and we're going to talk about that in a minute. Okay, so any questions about this? So this, we're not going to do much on this because the immunology is quite complex, but just recognize that you have random antibodies floating around in the blood and lymph, constantly checking cells for foreign to be able to bind. If they bind, they elicit a response. They activate the immune system, and the immune system takes care of the invaded cells. Okay?
Where did the mRNA spike come from?
They made it in bacteria.
And they put it into the cell.
So they make it in bacteria, they clean it, then they put it into these liquid droplets, these lipid droplets, okay, lipids like fat. Okay, and then that goes in the vial and gets dried down. Then at the vaccine place, they'd add saline to the vial, resuspend these droplets, and inject that into your arm, and then because it's lipid, just like your plasma membrane is made of lipids, this is able to bind to the plasma membrane and release the RNA into the cell.
Oh, I see. Thank you.
Sure. Any other questions? So basically, we're taking over, again, we're taking over the natural processes of translation to make the protein that we want to get an antibody against. Just like the virus takes over to make more virus. But hopefully, by getting people immune, we will prevent this because they will recognize the virus before it even gets to bind to a cell. Or if it does, it will limit it will limit your sickness because there's a lot of the antibody around already. So you may still be possible to get sick with being vaccinated, but it'll be a much shorter sickness and hopefully not as severe.
And one more question. The antigen was the protein?
Right. Antigen is the spike protein in this case because an antigen is what an antibody binds to. Okay. But spike protein also, of course, has a different function in the virus itself. Here we're saying, hey, this is what we want an antibody against. Like, let's go find it. And it really is a fishing. I mean, it's random. And so, but because so many antibodies are made, it's possible that it's already in your body.
So, like, if antibodies are like always random, say like the vaccine never happened, how long would it take for like a antibody to form for COVID, like on its own?
Right. So it's similar to so basically, right, with the antibody, what we're doing is we're activating the immune system without getting sick, right? Or you might get, okay, yeah, without getting sick. So you don't actually get COVID disease using the vaccination, okay? People who react to the vaccine and feel icky the next day, that's the immune system making them feel bad, not the vaccine itself, okay? The immune system, when it's activated, releases interferon, and interferon makes you feel like you have the flu. And like I like to tell